welcome everyone today to this Public Health Alliance for Genomic Epidemiology seminar. Today, Professor Colijn from Simon Fraser University in Canada is going to be giving a webinar on advanced genomic epidemiology for public health, novel clustering techniques, and serial interval estimation for SARS-CoV-2 sequences. A few kind of ground rules before we get into the introduction and I start the seminar. If you have questions during the talk, please use the Q&A feature. Any kind of clarifying, anything more short-term clarification, feel free to put it in the chat, and I'll jump in to clarify that. And then we'll have a 45-minute presentation, and then we'll have a 15-minute question and answer session at the end. So just to introduce Dr. Colijn, she's a professor, a Canada 150 chair at Simon Fraser University. Previously, she earned her undergraduate degrees from UBC before enrolling at York for a master's degree in environmental studies and completing a PhD at the University of Waterloo. Following that, she went on to do postdoctoral training at McGill University and at the Harvard Chan School of Public Health, and took up faculty positions both at the University of Bristol and then Imperial College in 2011. And 2017, as part of Canada's kind of drive to Canada's 150th anniversary and drive to bring back exceptional talent, Dr. Colijn was able to take up chair at Simon Fraser, was awarded a chair at Simon Fraser University, where she runs the Magpie group and has been doing some really excellent work throughout the pandemic, as well as beforehand, really driving the limits of phylogenetics and using phylogenetics to drive epidemiological insight in a meaningful way, leading directly to public health innovation. Please take it away, Professor Colijn.
Thank you. Thanks so much for the kind introduction and for being here. I know that some of you could be at APBHM instead, and I appreciate the audience. Okay, I will share screen and get started. So hopefully you can see this. Right, so this talk is in two parts. And the subtitle, first part is about novel clustering techniques and what does clustering really mean? And the second part is about how we use, going beyond clusters. What do clusters mean? And then is clustering enough, or do we actually want to be able to do something else with our sequences? So the first, I'll just get right in with the first part. So this is in the context, obviously of genomic epidemiology. And I guess throughout my interest in this area, one of the focal points for me has really been transmission. What can we learn about transmission? So sequencing is great. Sequencing can tell us about so many things. It can tell us about drug resistance. It can tell us about evolution. It can tell us about ancient history, in macro evolutionary context. It can tell us lots of things. But in genomic epi, I guess my focus has really been around transmission. So the context, as most of you are probably familiar, but since I don't know the audience, often sequencing is done on a routine basis or on a targeted basis. So people's pathogens are sampled, and whole genome sequencing or other sequencing tools are used. And then, the first task is often clustering. Group those sequences into closely related clusters for the purpose of learning, who's related to whom might have infected whom, what's an outbreak, defining an outbreak. And that in turn has a purpose, not of just knowing what the outbreak looks like, but of supporting epidemiological investigations and supporting a public health response. Okay, so that brings up some questions. How do we define clusters? Are they true? Are they meaningful units of analysis? Why and when would we expect there to be clusters? We kind of have this established tool, and I think it's rooted in certain contexts where sequencing, is done in places with a lot of resources, because it has been historically quite expensive, and much of the transmission is sort of elsewhere globally, somewhere far away. So what does that mean, about when we get these clusters, and how well defined do we expect them to be, and so on? Okay, sort of some intro context. So, all right, well, we like sequences, and one of the things we love about sequences is that we can make a phylogenetic tree, which draws a nice story and a picture of the evolution of and the shared ancestry of a set of organisms through time. So, and the tree is great, right? Because it can capture things like subtleties in the molecular evolution model. What do differences between these sequences really mean? Some differences mean more than others. At sites that have lower rates of variation, you might find more meaning in divergence at those sites than at highly polymorphic sites. Tree inference can deal with all of that and can sort of capture that information from a whole group of sequence. So why not do the following? Why not make a tree? And I made this one up and colored it in. There's established ways to do it, and they're great. And then define and identify clusters on the tree. And we do that. It's works largely. And here I've colored into clusters, take little clades and say, okay, everybody descending from this node, go, you're a cluster. So why don't we do that? And one answer is actually we do that. But there are some challenges. In practice, tree reconstruction can be computationally expensive. So in the pandemic in BC, they were sequencing about 4,000 or 5,000 SARS-CoV-2 sequences a week. So, after 10 weeks, you've got 50,000 sequences, and rebuilding a tree every time you want to rebuild clusters is computationally taxing. Furthermore, some of those sequences might be indistinguishable with the tools that you have, or they might really be the same. And so there might not be enough genetic diversity to create well-defined phylogenetic trees. And then you have, are you going to make Bayesian posterior collections of trees on your 50,000 sequences every week, with 55, 60, 65 next week? That's going to get intractable, once you get past 1,000 or 2. And then finally, given that context and the diversity challenges, the population structure can actually be spurious. And 2 cases that are clustered together this week might not actually be clustered together next week. And that's confusing and not useful for the purpose of driving and informing those onward epidemiological investigations. So on the right-hand side, I've just made in our tree, or in our COL, in our random genealogy with 20 taxa. There is no interesting population structure here, but it really looks like there's one kind of well-defined clade on the right-hand side of that tree, where there's interesting recent divergence and so on. But this is literally just what you'd expect at random from a coalescent model. Okay, so those are some challenges in practice with using trees to define clusters. I think tree methods are great when your tree really looks like this and it's well supported. But that may not happen in the context of closely related outbreaks that are going on in real time in infectious disease. Okay, so our challenges: we had sequences, we wanted to do phylogenetics and clustering, we wanted to inform outbreak analysis, those little pictures. But here were the aims. We wanted clustering that was feasible in real time, that was stable week to week, and ideally would reflect known epidemiological links in the context where we know that we don't know all the links, especially in a respiratory infection. People don't always know who they've contacted and so on. Okay. So here's a bit of background. This is up to mid-2021 when we were developing this work. We've obviously been using this tool since then, and the BCCDC, our collaborators, have been using this tool weekly still, but this is the kind of pandemic context until that time. So one of the interesting things in BC, let me move the little Zoom picture so I can read my own legend, was that there was a time in here when we had both high levels of gamma, P1, and alpha co-circulating at the same time. You can see that here. I guess the alphas are in brown, B117, and the gammas are P1 in green. So yeah, we had this whole phase here when we had a lot of co-circulation of both of these with quite different diversity patterns and different clustering patterns. Okay, so that's the context. And this is the number of sequences that we had. So you can see it gets up to the tens of thousands already by mid-2021. Okay, I've got my Zoom pictures. All right, so the fundamental clustering tool we have is pretty simple. We use a logit model, and we just say there's different ways that there's different sources of information that contribute to whether we think of 2 isolates as linked enough to be a link that helps define a cluster. And so we have logit P, and we have contributions from some offset, a genetic distance term, a time term, and then a whole bunch of other covariates could go in if you wanted. So that allows for, if you knew that there was shared exposure, shared location, shared household, and so on, you could put that into this model and you could develop a clustering that takes all of that into account. We didn't do that. We used genetic distance and time. We trained the model with known clusters, so to best capture the really high quality clusters that the Center for Disease Control here in BC, the BC CDC, had already defined. But also you can use expert knowledge of the mutation rate, the serial interval, the generation time, and so on to estimate these coefficients. So for example, if two cases are 200 days apart, that time signal does not drive them being, a link that kind of helps to define a cluster. If they're 2 days apart, maybe they do. Okay, so we called that method COV2 clusters, and what we did was compute this, so define these coefficients either with training or with expert knowledge, or in practice both, and then compute for every pair of sequences I and J, compute PIJ. That's the probability that they are linked. And by linked, we mean not in the same cluster. We mean close enough that the link helps to define the cluster. So we connect I and J if that PIJ is high enough, and then the clusters are the connected components of the resulting graph. So here are some little examples. In this example, the red and black edges don't really mean anything, but you can kind of see, yes, if you change the threshold and you start adding more red edges, these little clusters would merge to become one big cluster. All right, so far so good? Yeah, great. Okay, so then here's our clustering method. One of the nice things about this is it's very stable, right? Genetic distance doesn't change. If you add more sequences tomorrow, you don't change the distances between the sequences you already have. And so none of this changes. Nobody will get lost from clusters. And that was one of the challenges that they were facing in BC, was that with tree-based clustering, once you add more information, distances between I and J that you had before could actually change because the tree changes. And so you found yourself in a position of reporting, hey, that cluster that had 47 individuals last week, it now has 50, but that's 7 new ones and you lost 4. And that's really confusing. Okay, so this summarizes some of the results. That's the tree. These are pre-delta sequences. And so these are probably these are probably alpha and gamma up here. This one's probably alpha, this one's probably gamma. And you can see, oh, actually, no. Yeah, this one's going to be gamma, which is P1. Highly variable cluster size, lots of individuals not clustered. That's in gray. The panel here indicates clustering in the different COV2 clusters used with different thresholds to define the links. And we had some infeasibly large clusters. We did have a pretty good sensitivity for placing cases that had known links in the same cluster. So that's fine. Of course, it's the not in the same cluster that's always a challenge, because you don't know whether that's not in the same cluster, what the ground truth is. Okay, so as I mentioned, our clusters don't split apart when new data get added, so they don't have this problem that we were trying to solve, and we like that. And it's pretty feasible in real time, because as you add new sequences, you don't have to compute all the pairwise distances, you just have to compute the distances between your new sequences and anything that's going to plausibly have a reasonable score, and then recompute the clustering. Okay, this shows by week the number of cluster splits. So that's the number of clusters that actually broke up as you added more data. And you can see that the 2 tree cluster methods just have this problem quite generically, that every week there'd be clusters that used to be well-defined clusters, and then with new data they would split apart. Okay. I'm going to skip the entropy slide. I wanted to just highlight that, like, one of the challenges here in BC was, and probably elsewhere, was just times when there was very low genetic diversity, which led to all the sequences being grouped in one huge cluster. And that happened during gamma, and again during the AY25 delta, in the delta period in the AY25 sequences. So if there is actually really little genetic diversity, any clustering method is going to have trouble finding meaningful clusters that are useful, right? There just isn't the diversity there. And that, of course, led us to think about, what does clustering actually even mean? What do we want from it? It leads to some fundamental questions. So here are some, and I've got these Zoom pictures are really distracting. Sorry, I'm going to stop touching them and I'll put them down there and leave it. Okay. So clustering, of course, depends on the virus population dynamics. And if you think about it through the lens of coalescent theory, if you're not familiar with that, don't worry about it. If you are, maybe this will resonate. If you have a really small population and you sample N individuals and you say, well, how long do I have to go back until they share ancestry? The answer is not very long, because the chance, if the population is really small, the chance that two individuals have the same parent recently is high. So the population dynamics and the population size, and technically the effective population size, actually can impact the clustering patterns that you see. Another thing that impacts the clustering patterns that you see is what you're sampling. So actually, everyone gets their SARS-CoV-2 from someone else. It's not that it's living in a lake somewhere and we're all sometimes visiting this lake, right? So actually, A is connected to B, is connected to C, is connected to D. And sometimes when you have these apparent clusters, that's actually just because you're not sampling the missing links. So really, the cluster structure reflects how much of the transmission that's ancestral to your sample is actually in your jurisdiction where you're sampling, and how much sampling are you doing. So the epidemiology, the virus population dynamics, the virus diversification, how much transmission is happening per genetic divergence event, and how much you're sampling, all of these things actually connect to define what the cluster structure looks like. So when we say, oh, we have these clusters, this many percent of our, this many of our cases are in clusters, what that means is actually kind of unclear, because it's driven by all these factors. Okay, so for the second, so the first part is published as a paper called CoV-2 clusters, which just says the method and says it's stable. This is what we're doing. We're now about to submit a second paper on this, which kind of articulates what I just said. What does clustering really mean, and when is it useful? So of course, if transmission is really person to person, you have an obligate human pathogen. The truth is that there is one big cluster. That is the truth. Everyone gets their COVID from someone else. And that will work. You will uncover that truth in your genomic epidemiology analysis when transmission is happening where you're sampling, and you're sampling enough. And your links are defined in a way so as to capture transmission links. So these, throughout the rest of this part, which is maybe the next 5-ish minutes, this big red thing, the red here is the one truth when we have one big cluster. So we can do a simulation study to say, okay, well, let's look at the impact of how, changes in the transmission rate, the sampling proportion, and the genetic diversity, and kind of unpack these underlying causes of different clustering patterns. And this is work led by Ben Sobkowiak, who is in my group, and also with Ted Cohen's group at Yale. Okay, so when you have a high transmission rate, which means you have lots of transmission events per unit time, which means you have lots of transmission events per unit genetic diversity, right? Because that happens in usually a clock rate manner through time. Here's a simulation where we just have one jurisdiction and we have the truth. Everyone gets their COVID from somebody else. And what happens is we decrease sampling. Well, you see the truth, and then you see fewer and fewer are sampled. The big circles are sampled. What happens? Well, occasionally you see something that's actually diverse enough that it's not grouped in the same cluster, but basically with relatively low genetic divergence per case, we're pretty robust to sampling, and we uncover one huge cluster, which we conclude is not useful because it can't help you drive, who infected whom. But it happens to be true in this case. Okay, so what if the transmission rate is lower? Now we have fewer transmission events per unit time, and we can see what happens. We have the truth, we have 100% sampling, and we get our links are perfect in this little simulation, almost perfect. We actually simulate from our CoV-2 clusters model, or simulate, that's how we define our clustering. the transmission networks are sort of more chain-like and more time is going by per transmission. So what happens is as we get lower and lower sampling, we start to see what we call spurious breakup. So the truth is everyone gets their COVID from someone else in the jurisdiction, but actually when we sample, when we downsample and assign clusters, what we see is completely spurious, well, largely spurious. These folks are, these little yellow dots, I hope you can see the cursor, are more closely related to each other than everyone else, but not because there's something special epidemiologically going on, just because we missed links and so we called distinct clusters. Okay, so in spite of low sampling, most cases, get assigned to a single cluster at the high transmission rate. That's this one, and most of the cases are in this red blob. Then clustering kind of appears useful, but because it captures the true population structure, but the true population structure isn't useful because the truth is that everybody's in one big outbreak. In the lower transmission case, the clustering kind of appears useful, but it isn't useful in the sense of like really describing what's epidemiologically going on. It just appears that there are these different clusters and they're somewhat well separated. But if you started changing the threshold or the sampling rate, you would find that muddies the waters here because that there's no true population structure to be discovered. Okay, but of course, we don't have one introduction to one jurisdiction at a time. What we do have is multiple introductions seeded from a global pool that is in a jurisdiction that we're not sampling the rest of the world. So what happens? So I'll talk about high transmission and high diversity first, because this is the great case for genomic epi, probably the motivating case or one of the motivating cases, where we have high transmission in our sampled jurisdiction. So we have this is the truth, and each of these little colors is a different introduction from a global pool. And if we sample everybody and we do this perfectly, our clusters recover descendants of the distinct introductions from the global pool. When we downsample, we still mostly get that. So here clustering performs pretty well. What can happen though is that one of these with high transmission locally, one of these can be huge. And we saw that in the pandemic experience in BC, where some introductions just took off and had thousands of cases. And then of course, that's not particularly useful to say, oh, there's a cluster with 8,000 people in it. Okay, so what happens when we have lower diversity in the global pool? So this would be when Gamma first emerged, of course, and Omicron, it's brand new and it has low diversity globally. It's spreading rapidly around the world. Different introductions happen. They arrive in our jurisdiction. We sample them, we sequence them, and look, they all look the same. So even though we had different introductions, they actually all looked like one huge cluster. And that's how we ended up with 80% of our individuals in the same cluster. Okay, when you have low transmission in your local jurisdiction, what can happen is a combination of these previous things. You can end up with this phenomenon and some spurious breakup of the clusters because of lower sampling and longer transmission chains. All right, so I'm going to wrap up part one of the talk. But I think it's important because of the role that clustering tends to play to just note that, everyone gets their obligate human pathogen from someone else. So what cluster structure really means is there's transmission ancestral to your sample that happened among individuals you haven't sampled. So that's missing links in your jurisdiction, transmission outside your jurisdiction. And this can actually still be useful and correctly reflect something you really want to know. But it can fail. And it can fail by giving you one huge cluster that is spuriously joining different introductions because they all look the same. Or it can fail by spuriously breaking up clusters that really should be, nothing's interesting is going on. Everybody's getting their COVID from someone else in your jurisdiction, and you're just not sampling it very well. So I think we have to be really careful when we think about clustering to think about what's driving the clusters and what do we mean when we say there are these clusters. What do we think? How do we think these factors are playing out to define those? And then I think the next lesson learned is, clustering might not be perfect. It might not reflect the true population structure, and the true population structure might not be clusters. So if you don't, given that, what to do? is cluster membership enough? Is that what we want from genomic epidemiology? Obviously, because I'm here, it's talking about the next part of the talk. The answer, I think, is no. I don't think that's enough. I think we can and should do more with our surveillance data from a transmission point of view. I know we already do a lot with it, but from a transmission point of view, we should do more than grouping cases into clusters and talking about what percent is clustered and who's clustered with whom. So what is the more that we could do that's beyond clustering? So a lot of the work I did before the pandemic was actually in the first of these two major bullet points. And what I'm going to talk about today is the second one. But I wanted to kind of highlight that this first area exists and we love it and we do it. Can we reconstruct stories of who infected whom, capturing uncertainty with whole genome sequence data? And I think sometimes we can. You need enough diversity. You need sometimes you need additional information. But if you can do that, you can think about determinants of transmission. You can think about spatial roles of transmission. You can think about the timing of transmission. We looked at, before or after people had moved to Norway in one paper. We looked at before or after clinical presentation of symptoms of TB in another paper. So that's all useful information. And even if there's a lot of uncertainty, you can still get that out. Now, another thing motivated a little more by the COVID context is estimating serial intervals. And that's what I'm going to talk about next. Okay, so we have new methods to use sequence data to estimate the serial interval distribution. And these are designed for settings where there's not enough diversity to really unpack like all these stories of who infected whom and all that stuff in the first bullet point. So what's the serial interval? It is the time between symptom onset in person A and in person B if A infected B. If they don't have symptoms or symptom onset data is not reliable, in practice, we've used sample collection date in A and B where A infected B. These can actually be negative. It could be that A infected B and then B gets symptoms first, but we're going to forget about that for this talk and imagine that we understand the direction of transmission from the timing of sampling or the timing of symptoms. So why do we care about these? These are actually fundamental to infectious disease modeling. And infectious disease modeling is used for all the stuff about forecasting scenarios, vaccination, rollout predictions, how many people you might have to vaccinate, where is your outbreak going to go? So R0 and RT. And serial intervals also inform how fast we need to intervene to stop transmission chains. If you think that it's going to take only 2 days on average for symptoms in one and symptoms in the next, that informs you about the average generation time, which is in onset of infectiousness in someone and infecting another. And that in turn helps us think about how fast we need to intervene. So they're actually pretty fundamental, and usually they're hard to estimate. I'll flag this work is led by Jessica Stockdale, who's a faculty member in our group and was a postdoc in my group. Okay, so how do you do this? Well, conventionally, you have to find pairs where you're really confident that A infected B, and then look how long it took, and then do statistics to adjust for missing information and truncation and so on. But the first step, finding pairs where you really think A infected B, is hard. And that takes a lot of boots on the ground epidemiology. And it's data that's very hard to share because it's personal data, it's contact data, it's individual data. And it's often not available. And it's often actually not really systematically collected in a format that is amenable to kind of passing it out to researchers and doing this estimate for modelers. Often contact tracing teams don't need modeling estimates. And so there's lots of barriers there. Our insight was to ask the question, could we use virus sequences as kind of a proxy for contact data, knowing that they don't tell us who infected whom, but they do tell us something. They tell us whether A and B are a plausible pair. So in our model, if A was observed first or had symptoms first, then A might have infected B, but maybe that's indirect and we understand that we don't know because we understand that we have many very similar sequences. But we did find that this enables us to pull out some information about serial intervals right from virus sequence data. And that's potentially really powerful because it can be outbreak specific, context specific, variant specific, and doable with routine data that in a way that we can't, get these estimates from standard methods, right? Because we can't access that level of personal data. Okay, so we worked in collaboration with some researchers at Melbourne, Courtney Lane, Ben Howden, and Anne Watt, among others. And we had Melbourne, kind of wave 1 and wave 2 outbreaks from 2020. That's in March, and that's in the summer of 2020. That shows you a phylogeny from wave 1, and the cluster patterns are kind of drawn onto this phylogeny. So these are the different clusters that are illustrated here. So that shows you a little bit about the data. These are SARS-CoV-2. Okay, and I'm going to talk about the method. So we started with these clusters, and in wave 2, we actually started with exposure sites, and we used different information to kind of define our units of analysis. We identified these plausible pairs. We sampled lots of possible transmission trees because we don't know who infected whom. Each one we do our estimation, which I'll tell you about, and then we average those parameters over all of those trees. So it incorporates a lot of uncertainty. So there's the pipeline schematic. Sequence sampling and sequencing happens. Somebody else in our collaborators do this. Then clusters or exposure sites, cloud of possible transmission networks, and then our estimation captures the possibilities for direct, indirect, and co-primary transmission. All right, so what are those? Well, we know that we may not have observed everyone, right? Because we know we don't always sample everyone. I should say this method is, best where you have sampling enough that you really think you do have some closely related pairs. But we've modeled 3 things that can happen. You can have someone directly infecting someone else, where both of these red schematic individuals are in your sample. You might have some indirect unsampled people here illustrated in gray that are in between. So A infected X, all these unknowns, and they infected B. Or it could be that actually A and B look like they might be a pair, but they aren't really a pair. They're both infected by the same unknown individual. So we have these 3 things contribute to our statistical model. We want to estimate the time between A and B in this direct route. So we assume that's a gamma distributed thing, has to be positive. That's a limitation. It might be indirect, in which case, if this is gamma, then this is a sum of gamma random variables, and it's a sum of a geometric number of gamma random variables, because we don't know how many of these gray people we have. That gives us a compound geometric gamma distribution for this route. And actually, this is the same as this, right? The direct route is the same as this, but just with 0 gray people in between. And then we have this other route, which is the gray individual infects both. And here, the time between the red and the other red is described by a gamma difference distribution. So this GDD. So that gives us a statistical model, which is a description of the distribution for the time between the 2 red people, TIJ. So TIJ is either from this compound geometric gamma with some mixture fraction W, so that's the first 2 roots, or it's from this gamma difference distribution with the rest of the mixture. And what we do is we get all these TIJs from our plausible pairs from our genomes, and then estimate the 4 parameters of this model using, but you can use maximum likelihood. We used a beta prior on these 2 unknown parameters, and then we used a kind of Bayesian approach. Okay, so plausible pair, what does that mean? Of course, people might have more than one possible infector. So how do we handle all this uncertainty around the who's in a pair with whom? What TIJs do we actually pull out of our cases and sequences? Well, we say that 2 people who are close enough in symptom onset time or collection date, and whose viruses are close enough genetically are a plausible pair. So if you're 30 SNPs apart, you're not a plausible pair for COVID transmission. If you're 0 or 1 or 2 SNPs apart, you probably are a plausible pair, and maybe it's more likely with 0 SNPs. Similarly, if you're 8 weeks apart, you're not a plausible pair. If you're a week or 2 or maybe 3 apart, you are a plausible pair with like a higher probability for somewhat shorter times. So we use some information, of course, in defining that model, the information that we know about how SARS-CoV-2 works. Then for every individual, we select 1 among the possible infectors, except for I guess we have possible index cases here. And then we use this approach to create many, many trees, and we use all of them because they're all plausible. Okay, so then we estimate a likelihood, which is the same gamma difference distribution. And I've already said this. I guess the important thing here is really that we have uncertainty that arises within each tree, and then uncertainty that arises because we have many possible trees, by which I mean transmission trees of who infected whom trees. Okay, so that's some of the sampled trees and graphs, what they look like. And I won't talk about this too much. I'll just leave this here to say these dashed lines here are the truth. And this is from a simulation study where we simulated inter-onset times from our mixture model and asked, can we recover the truth? So the mean that we estimate, how different is it from the true mean? It goes to 0. And how does that depend on the number of samples and on how variable the actual distribution is? So if the serial interval is 10 days plus or minus half a day, then you go, okay, it's like 10 days, 10 days, 10 days. It would never be that good, right? If it's 5 days plus or minus 5 days, it's kind of going to be a mess because people could infect each other after one day, or you could have all these different routes. So this kind of shows that as we get to a mean and more data, that we do actually converge on the truth. Okay. Here are some estimates from Melbourne's first wave, and this is the mean serial interval, and you can see we get some variation between the clusters, but it's not significant. And we get some variation between the serial interval, the standard deviations. But we do see some cluster-specific estimates, and we get serial intervals that are like really in line with published estimates. So that's great. I think I have a better slide for this. Yeah, here it is. So this is for Melbourne's Wave 2 data, and here we actually have some information about what the clusters, what kinds of settings the clusters were in. And you can just see them colored here where we've colored the different categories. We don't see a lot of like, oh, look, these ones are all shorter than those ones. But we did see kind of meat packing and processing plants tended to be, and schools tended to be on the shorter end. So you can see orange down here, here's the packing, meat processing, and you don't actually see any orange up here where it's longer. Healthcare facilities and housing tend to be on the longer end, and we do have a lot of variation. So with mean serial intervals ranging from 2 to 10 days. I think the jury's out. This is kind of not probably very significant in statistically. If you test like, hey, are meat packing different from everybody else, may be different from some of these longer ones, but it's not a super clear signal, and that's okay. I note that serial intervals do change during an outbreak. So if you stop an outbreak early before serial intervals start to contract, you might get longer serial intervals. That's one reason these might look longer, is they were actually stopped earlier. But I don't know what the drivers are of these differences. Okay, so I motivated the serial intervals by saying like, hey, these matter for modeling. They matter for transmission dynamics. They're this fundamental parameter. So, okay, do they matter in this context? What we show here is what our serial intervals mean for the parameter R, which is the effective reproduction number during these outbreaks through time. So each panel is an outbreak or a cluster. And the estimates shown is RT over time during that period. And mostly our clusters and the standard estimates would completely overlap. So we don't actually see a huge difference. If you look at our estimate of the serial intervals as an input for this estimate of R compared to using a standard estimate for the serial interval from the literature. But sometimes it makes a big difference. And I've highlighted those with these big black arrows. So here in this cluster B47, the estimate for R that we would get is only 2, whereas if you used standard methods to standard serial intervals, you'd get 4. That makes a big difference to how much transmission you think is happening in the first stage of this outbreak. Similarly here, our estimate is half what it would have been. So it impacts us. If you were going to derive onward modeling from these outbreaks, you would say, oh, look, R is 4, we have to model that. You'd get a much more pessimistic view than we have, and that's because an input to these R's is the serial interval. Okay, we compared this to using contact data to define the pairs instead of using genomes, and that's hard because the clusters don't exactly align, but we do get fairly similar results. And I'll just end with this one. These are published estimates of the serial intervals for COVID, and these are our estimates, and you can see that our estimates are much more wide, and I've highlighted two kind of opposing reasons for this. One is that published estimates have better data, right? These are data that do have boots on the ground intensive epi investigations and very rich contact data. So they might have better transmission pairs, and that's going to reduce the noise and the variability. They also often don't account for the idea that you might be missing anybody. They say, these are our pairs, we know them for sure. Two people went to the same place at the same time. They both got infected. One infected the other. It could have been that they actually both were infected by a third. So that co-primary route, that missing infector of both the red individuals, that actually drives uncertainty. And I think that should probably be taken into account. But largely our results are, they're not wrong. They're just more uncertain, which is to be expected. Okay, so conclusions for the talk. Clustering is great. It's really great. But it's not always right. Sometimes it's wrong. And I don't think it should be the end point. I think it's a starting point. It can fail to reflect true population structure, or it can reflect true population structure, and there can be not much clustering, and that can be truth. So we can do more. We can estimate serial intervals. We can reconstruct transmission networks. And if we have these estimates, we can actually do onward modeling. So we could think, like, what's the force of infection from different groups? Once we have these plausible pairs, we could think, could we estimate, the contribution to transmission of breakthrough infections, for example? So that's a direction that we're going to move in our group, is like using this work to inform models, because the models that are used for forecasting and prediction tend to be really simple and are not informed by this rich genomic data that we have from genomic epi. So I'll just end with, if you have data and you think these topics are interesting, feel free to get in touch, because we'd love to hear from you. And I want to thank especially Ben and Jessica, whose work I highlighted, and our collaborators who provided data and interpretation from both Melbourne and BC. And that's it. Thank you.
Thank you very much. Really interesting talk. I always enjoy seeing the limitations of some of the methods, and really love the way you looked at the robustness of all those inferences. So we've got a few questions, and if anyone wants to ask any questions, throw them into the Q&A panel. So Dr. Soin Kim asks, would the impact of downsampling be different with different clustering methods? So tree instead of the logic model.
That is a good question. So in the simulation study, one of the things we looked at was the impact of downsampling on the truth. So assuming your clustering is actually working perfectly, like what happens when you just downsample? So I guess the differences between the different methods would come from how well those different methods actually perform. So if a method is based on a tree, and downsampling actually means your tree is a lot more stable because you're not changing it every week, because you're not adding much more information, then that would actually reduce the instability, but it might still lead to the spurious breakup.
Did you find the logic model needed to be retrained over time, as things like your heterotachy and your Omicron obviously messing up everything and messing up all the thresholds, or could you just compensate by changing the P threshold you were using?
Yeah, so what we found, we did play around with that, and there's obviously some trade-offs, like you can achieve the same thing by changing the coefficients or changing the P. So in a sense, it kind of doesn't matter which one of those you do. What we found was that the one huge cluster problem does not get fixed when you change the coefficients or the P threshold. It does change the edge case. So you might have, instead of 80% of your cases in one huge cluster, you might have 72% of them in one huge cluster, and another 8% that are unclustered or are in really small clusters. But what it never did was take that 80% in one huge cluster and divide it up beautifully into 30 equally sized, like, tractable clusters. And that's, I think, a sign that, like, the truth really is just one big cluster, whatever your parameters are. But yeah, we did tweak it throughout the pandemic. And that was partly the need to do that, was partly what drove us to start asking these questions. Like, why is this not working? What would we expect to see here? Would we expect this to work? And so on.
So Dr. Kim asks again, kind of a couple of questions kind of aimed at more SES factors in relation to these intervals estimations. So what was that a consideration in any of your analysis? Would your findings differ if some of these social factors were incorporated? And do you expect R to be different depending on social status?
So yeah, that's a great question. And I guess I'll use it to pitch something I really care about, which is connecting additional data or metadata to our genomic epi. We never had those data. So I wouldn't, I can't answer that question. And I think there's reasons for not sharing some data because it might be stigmatizing. But on the other hand, you can't figure out if different groups are being treated differently if you don't have those data. I would not expect, there to be differences that are because of those social and economic factors directly. I don't think certain ethnicities or whatever transmit COVID faster than others but I would expect that different epidemiological contexts would have different epidemiological dynamics and patterns. And of course, you might have a different composition of a workforce in a meatpacking plant than in a shopping mall or in different outbreaks. And it does matter. And I think it matters for, if we have a policy or a practice around intervention or prevention that is based on an assumption that serial intervals are all the same, and they're not in a way that disadvantages groups that are overrepresented in slaughterhouses, for example, or meatpacking or other industries, then we need to think about that. And there's an added value to thinking about this more carefully because it has equity considerations.
I don't think if I may disagreement with missed opportunities in the genomic and the epi data linkage within Canada, especially. In terms of one of the areas of clustering that increasingly is coming up in clinical data sets that I'm dealing with is subgenomic clustering. So the perils of mobile genetic elements and trying to infer linkage and transmission in mobile elements, which in turn tend to be associated with mobile elements. So you have a single gene with low diversity, but within a small genomic context, potentially a lot of diversity.
Right.
So how do you even go about trying to link that without doing a manual analysis every time?
So, right. Yeah, I'm not sure I understand the question well enough to answer, but so you have a mobile genetic element and it's sitting in a context of broader diversity, but it's moving around and you can start to transmission through that.
So an example would be you've got multiple individuals that have within a hospital that have all tested positive for extended spectrum beta lactamases. And then the question is, you've sequenced the genomes, are these actually a linked mobile genetic element related transmission of that resistance gene? The problem is the gene's on a plasmid, but then it's also on another mobile genetic element within the plasmid. So the plasmids can be different, but then the transposon might be the same, but there's just a couple of layers of different ranges of diversity in a very small area. So just trying to go about ways in which you would cluster that kind of data that can handle a range of diversity within a small distance, essentially.
Yeah, that is a cool question. I guess I would, and then this is just like off the cuff, obviously, so happy to talk more, but I would start by defining a set of different distances that you could use because you're looking at the same gene, but you're looking at, these different views and actually seeing what the cluster pattern looks like with all of them. And I guess it's plausible to think that if any of them really give you a consistent picture of a linked outbreak, that's probably the truth, right? Because it seems like the probability of getting all these cases spuriously linked, if they're not, is very low. Whereas the probability of looking at the wrong distance measure and getting them spuriously not linked if they are is pretty high. So you have kind of a asymmetry there, which maybe you can use.
Yeah. I think Dr. Wilsow has some fun data related to this as well, locally, if you want to chase it for that. But yeah, it's an interesting problem that is. I'm just curious to see how you could try and expand the same approach for the relatively well-behaved SARS-CoV-2 genomes in the grand scheme of pathogen genomics
Right.
to
I mean,
that
I
kind of
think
more
you,
complex sense.
yeah, you might also find a lot of these same things. Like if you do have really rapid transmission, you'll have low diversity per case and you'll get one huge cluster. But in that context, the one huge cluster may not be your challenge. The challenge may be failure to detect genuine links. And so then you want to increase your sampling, which is expensive.
Yeah. I think one last question from the chat. What about host pathogen interaction? Say less microbiome diversity in low income populations. So
Yeah,
the kind of high.
Yeah, that's, we have not worked on that. Like I said, we never have data about the income level of the, cases corresponding to samples in genomic epi. We do have that data. So in some of our TB analyses, we have more metadata. So around a history of homelessness or alcohol use or past treatment history. But we don't also have microbiome diversity data. So, and I think the challenges would be a little bit different. Like, I think that's really more a question of, let's think about what's happening in microbiomes rather than let's think about who's linked to whom and what the transmission chains are. So quite
I suspect that kind of analysis probably more limited things like some of the MRSA, bigger, the big Broad Institute scale MRSA analyses.
Yeah. And even there, the focus isn't really transmission. The focus is more the biology of the microbiome.
Yeah, and colonization protectivity.
Yeah.
Okay, I think that is, unless anyone has any last minute burning questions in the chat. Gonna take that silence and say thank you very much for a really interesting seminar. I really enjoyed your treatment of these complex, seemingly simple, but in reality incredibly complex things that we've been trying to do for the last several years.
Yeah, well, thank you. It's great to meet you and great to have done this. So thanks very much.
Thanks, everyone. Have a good day.
Okay, you too. Thanks all.
Bye.